Now for the next and our last topic for this webinar, the non-replicating vaccines. So, as we all know, viruses need to invade host cells to survive and replicate by hijacking the protein-making machinery so it read the virus genetic code and make new viruses. And these virus particles contain antigens that can trigger an immune response, then we become infected. The aim of our vaccines, they will expose our body to an antigen that can't cause disease but will provoke an immune response that can block or kill the virus if we become infected. So all of the vaccines work the same way, but the method of exposure is different. And in the case of COVID-19, when it was discovered that it was a coronavirus, namely SARS-CoV-2, and since there are previous research already on SARS-1 and MERS, researchers knew that they could focus initially on the S protein, which is also known as the spike protein, that is found in the surface of the SARS-CoV-2, which helps the virus penetrate the human cells and initiate infection. And inside the cell, there is a genetic material that contains instruction for making proteins. And it also contains spike protein gene that instructs the virus to make the spike protein. The scientific explanation for this is that the spike is a target antigen, and that's the strategy behind most of the vaccine candidates for COVID-19. Now let's have our types of vaccines. So this was already discussed by our first presenter, but let's just have a short recap about this. So the types of vaccines comes with different approaches. There are two classic approaches of vaccines that we are using before and until present, where the virus is either weakened, that is live attenuated virus, or killed, the whole inactivated virus. And since there are huge advancements in vaccinology, it had opened multiple other approaches to developing vaccines where there is no need in handling actual virus. So what we need to have is just a sequence coding of the virus like the conventional approaches, which includes the virus-like particles and subunit vaccines, and the so-called next generation approach. So this includes the genetic vaccines, which has two types, the RNA vaccines and the DNA vaccines, and our viral vector vaccines, where our topic non-replicating vaccine type belongs together with the replicating type. But before we proceed to our main topic, let's discuss first what is this viral vector vaccines and how do they work. So viral vector vaccines are newer technology using harmless viruses to deliver the genetic code of the antigen to the cells of our bodies so that they can produce protein antigens to stimulate an immune response. The basic idea for this vaccine is the sequence coding for an antigen is inserted into different virus, and this virus use is attenuated or weakened so that it can infect someone without causing disease. So this virus is safe, and it serves as a platform or vector to deliver the protein antigen. And for this type, some vectors replicate in the body and some do not. So how does this vaccine type work? Let's have the SARS-CoV-2 as an example. As what I have said earlier, SARS-CoV-2 virus contains a gene which makes its spike protein. Scientists have identified this gene and modified other viruses so that it can be inserted into it, and that is our viral vector. Vectors have been modified to ensure safe gene transfer without inducing pathogenic effects or it can cause disease. So all of the viruses can serve as vectors once their replicative and pathogenic potential is removed and the gene of interest is inserted within their genetic material. So as you can see in this picture, this is an adenovirus vector where inside there is the gene of the spike protein. And then our vaccine will have these viral vector particles for this vaccine type. So our vaccines now contain these viral vector particles, and once inside our cells, the virus spike protein will be produced and then triggers an immune response in our bodies. So there are two types of viral vector vaccines: the replicating and non-replicating. First, we have the replicating viral vector vaccine. From the word itself, replicating, it produces new viral particles in the cells they infect, which then go on to infect new cells and triggers immune response. Because replicating viral vectors retain the ability to make new viral particles, it can provide a continuous source of vaccine antigen over an extended period of time compared to non-replicating vaccines, and so is likely to produce a stronger immune response. An example of this type is a vaccine to prevent Ebola called Ervivo, which uses a recombinant vesicular stomatitis virus. And this vaccine was approved across Europe for use in 2019 and has been used in multiple Ebola outbreaks. So far, this is the only viral vector vaccine that has gotten FDA approval. Now we have the non-replicating type of delivering vaccine. This type do not retain the ability to make new viral particles, unlike the replicating viral vector vaccine. This is because key viral genes that enable the virus to replicate have been removed. This is the advantage that the vaccine cannot cause disease and adverse events associated with viral vector replication. However, vaccine antigen can only be produced as long as the initial vaccine remains in infected cells. So it will just last for a few days. This means that the immune response is generally weaker than with the replicating vectors, and booster doses are likely to be required. So these are the viruses that are being used as vectors: the vaccinia virus vectors, adenovirus vectors, adeno-associated virus vectors, retrovirus vectors, lentivirus vectors, cytomegalovirus vectors, and Sendai virus vectors. Among them, the vaccinia virus vectors and adenovirus vectors are the most widely used vectors because they can induce a robust immune response. Generally, viral vectors achieve high immunogenicity without an adjuvant, unlike other vaccines, because viral components itself stimulate the innate immune response, leading to the production of interferons and inflammatory cytokines. To date, several adenovirus-based vaccines are in clinical and preclinical trials. Meanwhile, the most common among the COVID-19 vaccine candidates are adenoviruses. So here are the non-replicating adenovirus-based SARS-CoV-2 vaccine that are under clinical trials as of April 6, 2021. Some of the COVID-19 vaccine candidates use human adenovirus viral vectors. This includes the Russian Sputnik vaccine and the Chinese CanSino Biologic vaccine. One of the potential issues with these vectors is that inevitably some of us will have been exposed to these viruses before. Because of this, we may have some degree of immunity to them. This means that the viral vector itself produces an immune response, which may mean that the immune response to SARS-CoV-2 isn't that effective. So we have the Russian Sputnik V vaccine from Gamaleya Research Institute, which is already on phase 3 clinical trial. The viral vector or technology used is the human adenovirus vector, replication of AD5 vectored and AD26 vectored vaccine expressing the gene for SARS-CoV-2 full-length glycoprotein S. Phase 1 and Phase 2 studies showed both 82.6 and 85 formulations were safe and well tolerated. Combined regimen was better than the individual 82.6 or 85 injection. No serious adverse events were reported. The phase 3 study showed a protective efficacy of 91.6% against COVID-19. Next, we have the Kodvidisha from Cancino Biologicals Incorporated, Beijing Institute of Biotechnology, which is on phase 3 clinical trial. The viral vector or technology used is a human adenovirus vector, replication defective AD5 vectored vaccine expressing the spike glycoprotein of the SARS-CoV-2. The phase 3 trial are being performed globally with 40,000 participants, and it is expected to be completed by January 2022. Next, we have the Johnson vaccine from the Johnson Pharmaceutical Companies, which on phase 3 clinical trial, the viral vector or technology used is the human adenovirus vector, replication incompetent adenovirus serotype 26, vector encoding full-length SARS-CoV-2 spike protein. Early animal studies showed promising efficacy with low-dose single-shot vaccination. Subgroup analysis based on regions showed a higher vaccine efficacy in North America compared with South Africa and Latin America. This may be related to the difference in prevalence of mutant strains of SARS-CoV-2 in different regions. And now we have the ChAdOx1 and COVID-19, or the AZD122, which was developed by the University of Oxford and AstraZeneca. Currently, this is on phase 4 clinical trial, which uses a viral vector chimpanzee adenovirus vector vaccine with SARS-CoV-2 spike glycoprotein. In this vaccine, a modified version of chimpanzee adenovirus is used. The vaccine was given emergency authorization by the United Kingdom in December 2020 of the pandemic, and India authorized a variation of it the same month. As of February 2021, versions of the vaccine produced in India and South Korea gained approval for emergency use by the World Health Organization, which should improve accessibility and ease of approval for other nations. So this vaccine, ChAdOx1 and COVID-19, appears to be better tolerated in older adults than in younger adults, and it provides similar immunogenicity across all age group after a booster dose. In terms of safety profile of this vaccine, there are 13 serious adverse events occurred, but none was considered related to either study vaccine as assessed by the researchers. All the viral vector vaccine candidates for COVID-19 are non-replicating. This means that they do not create additional viral vectors in the cells that they infect. Though they need higher doses than replicating viral vector vaccines, it also adds to our confidence in their safety. There are a handful of vaccines available for clinical trials in the treatment of COVID-19, and these newer technologies, such as these viral vector vaccines, the non-replicating vaccines, showed a promising results. However, questions have been raised concerning the efficacy against new variant strains, because other countries showed secondary immunity alone was not sufficient to arrest transmission. So it's early to say which among these approaches of vaccines, the classic, conventional, or the next generation approach, is more safe and effective. That's why further rigorous studies and clinical trials are required to test for the efficacy of existing vaccines against mutant variants. So this is the end of our webinar for our Activity number 7 in new drug development, giving you information about mRNA vaccines and non-replicating vaccines. Again, this is Group 1. In behalf of my group mates, thank you everyone for listening. God bless and stay safe.